Yeah.
Through all the variables, you might.
There's one. Eduardo.
Should be. If not, oh well. It's too late now.
Oh, he said he got it.
Good one.
It works. She said it works.
Yeah. All right. Am I standing in the right spot?
Yeah.
I'm gonna sit there. Okay. All right. Thanks everybody for coming for Luke Powers, PhD dissertation defense. Glad you made it on this holiday. Just a few words to introduce Luke, a little of his background. Luke was born in South Africa, but grew up in South Carolina. He got his bachelor's degree at Erskine College in South Carolina, his master's degree at Southeast Louisiana University, where he worked with Kyle Pillar. Luke came highly recommended. He had several awards and fellowships at Southeast Louisiana, and that trend continued here. I don't know, there's too many to list. NSF Dissertation Improvement Grant, Tom Slick Fellowship, the Texas Chapter American Fisheries Society gave him a scholarship, and so on. Luke came with a pretty well planned out goal for the kind of research he wanted to do. He wanted to look at fish community structure, but he wanted to do like a broad global study. So I thought, well, that would be cool, but how are you going to travel around the world? Well, fortunately, he got the NSF grant, so that helped a little, and then I helped him with some of the other travel. But he pulled it off. To his credit, he got to several locations throughout the world to do this comparative analysis. But I'm going to let him share with you now.
And thank you, Dr. Weinmiller, and thank you for coming on this Good Friday, a half day, to watch my dissertation defense. So a famous ecologist, Robert MacArthur, stated that the general goal of all science should be to search for general patterns and rules. And this applies to physics, chemistry, and biology as well. And in community ecology is no exception. And in community ecology, we're trying to find these general patterns of community assembly processes. And this can be defined as the processes that shape the identity and abundance of species within ecological communities. And if you've ever studied ecology, you know this is pretty difficult. There's always some exception to the rule, some species doing something weird or different that you don't expect. And this drove people like Lawton to say that community ecology is a mess, and that it's just too variable and too complex to be able to find these general patterns or rules. But there may be hope. McGill and others have said that functional traits may be a way to find these general patterns. And functional traits are just any kind of morphological, physiological trait that can influence where or how a species interacts with its environment. An example of this can be the bill shape of a bird and how it relates to where it feeds. So, for example, this hummingbird has a very narrow beak, and so it tends to feed on the nectars and flowers. Whereas this blue heron has another bill shape that's better for catching fish and frogs, and not so good at getting nectar out of flowers. And so these traits really mediate species interaction with the environments. And this can help us get a mechanistic understanding of the species trait or species environmental relationship. And you can also use functional traits to look at different niche dimensions. So some traits can relate to different niche dimensions. And so you can look at individual niche dimensions as well as multiple niche dimensions. And this can really improve our understanding of species coexistence. And it can facilitate comparisons across regions. So rather than just focusing on a taxonomic unit, you can compare it across different assemblages and just focus on the function rather than a taxonomic unit. And so in this dissertation, I have 3 data chapters, and I'll be talking about each 3 of them today. The first 2 focus on community or convergent evolution and how that may be used to find general patterns. And then the last chapter focuses on functional and phylogenetic structure and what patterns that can show us globally. And so today I'm going to first go over some methods that are consistent throughout all the 3 chapters and then talk about each chapter individually. So as Kirk said, I went all over the place to collect data. So I collect data from the United States, Belize, Brazil, Benin, and Cambodia. And within the United States, I went to Texas, South Carolina, and New Jersey. And so at each of those locations, I sampled roughly 6 streams, and I tried to sample at 3 different scales: the microhabitat, a stream reach, and the whole zoogeographic region. So one of the things that was probably the hardest about this project was trying to find consistent streams, and streams that are very similar around all these areas. And so I found streams that always had sandy bottoms. They had to be forested. They had to have as little human impact as possible, and they had to be low gradient, so not mountainous streams, low flow streams. And here's just a picture of the stream from Belize. And then each of these stream sites I collected from 200 to 500 meters, and then within that stream reach of 200 to 500 meters, I collected microhabitats, and these are just consistent areas of substrate depth and water velocity. So if this is a stream, what I would do is I'd go along and I'd sample different microhabitats, so like maybe this loggy region or this sandy pool, and then I would collect all those fish and put them in a bag and flag where I got the fish from and come back and measure the substrate depth and flow. So I knew that those environmental factors for each individual fish that I caught. And then for every species, and for 5 individuals for every species, I measured 30 functional traits. I standardized them by length. And I broke these up into 3 trait categories. So traits associated with habitat use, traits associated with diet, and then traits associated, or all traits combined, including life history categories. I got a tree from the literature, a recent tree from the literature, and I calculated the phylogenetic signal for both the traits and the niche position of the habitat preference of all these species. And so here's just a basic summary of how many species I caught and a breakdown of the species per country. And so I got about 230 species, and I sampled 38 different streams. So my first chapter really focused on the similarity of environmental trait relationships in each of these countries. And so I was hoping to see if you had these similar relationships that I would suggest some kind of key underlying mechanism that's repeated and influencing these species traits in a similar manner around the world. And so this is based on the habitat template theory, which is the idea that the habitat creates a template or restricts the traits that can occupy a certain habitat. For example, you have this desert habitat, the plants from this coast-region jungle will not be able to persist or inhabit this desert because it doesn't have the correct traits to be able to persist there for long or live there. And so the idea is if this pattern is truly, if this is a general pattern, you'll be able to see this no matter the geographic location or evolutionary lineage. And so what you should be able to see is this trait environmental relationship having it repeated in different locations if A and B are 2 different regions. And there was a study, I got this idea from a study that was done in 2002. And so this is just percent is just a trait, and this FR is the flow. And so they saw a negative relationship between this trait and flow for 2 different regions, for France and Virginia. However, a lot of these past studies have focused on just a few regions, like just the United States and Europe. And really to find general patterns, you should be trying to do this in more geographic locations and different evolutionary lineages. And that unfortunately requires huge and large data sets. And oftentimes, just using other people's studies, there's inconsistent methods, whether it's how you choose the sampling sites, the scale you use. My microhabitat may be different from the microhabitat that you look at, and also different ways they measure traits or how they analyze the traits. And recently there's a paper, and it's actually maybe had to redo a lot of my third chapter, or one of my chapters, or this chapter, was that using these regression-like analysis to see the relationship between traits and environment tended to increase this type 1 error, and that you should rather use this R2L and 4-quadrant analysis, and which I ended up doing. And also, another problem is that a lot of these studies use very broad categories, so small, medium, and large body size rather than continuous traits. And so I'm going to try to address all these issues by sampling consistently in all these regions, and use not categorical variables. So the questions for this study, my hypothesis was that the pattern of trait environmental relationships will converge across distinct biogeographic regions as a result of common sets of environmental filters acting on functional traits. And so to answer this, I really had to ask three questions. Is there really a relationship between these traits and the environmental variables? And are these consistently seen across all these geographic regions? And is this really because of a common ancestry, or is this an adaptation to some similar selective pressure? And so you see convergence. So again, I'm sampling at all these different locations, and the scale that I'm really focused on is microhabitat for this particular chapter. And so looking at that, this hypothesis, I'm using these proposed methods of RQL and fourth corner by Ernst et al. And Dre. And these analyses let you see the relationship between the environment and the traits. And so looking at that first question, is there actually a relationship between the environments and the traits? And so this is a plot of that fourth corner analysis where you have traits on the y and the environmental variables in x, and a significant negative relationship is shown in light blue and a significant positive relationship is shown in red. And I just wanted to show you that you do see many significant relationships between traits and the environment. So there is this relationship. And if you plot just the traits with the environmental variables, here's body depth on the y-axis and velocity on the x, we do see this a relationship. At roughly about 0.2 meters per second, you see this loss of these deep-bodied fish, and this reduction in trait variation as you increase water velocity. And this can be seen in other traits. As you increase water velocity, you tend to reduce the variation in traits that you see. And so the more interesting question is, are these patterns repeated across all these different regions? And this is a lot to look at. So again, this is these are significant negative, these are significant positive. But what we're trying to look at is, are there similarities across all these regions? And I only found one relationship, and that's between body width and root bank. And root banks are just these roots coming from the side of the bank and where fish tend to inhabit and occupy these areas. And I was really hoping to see more consistent relationships, but I didn't. And one I was really expecting is that relationship between body depth and water velocity, but I only saw that in 3 out of the 5 countries. And this is just a global combination of all the data sets. But when we do plot this body depth by water velocity, we tend to see this general trend that once you hit these higher water velocities, you tend to lose these deep-bodied fish. And this is consistent throughout all the regions, and even in the global combined data set. And this may be due because of the amount of energy it takes for fish to inhabit these areas. If you have a deep-bodied fish, they're going to have to swim a lot and use a lot of energy to maintain their position in these high-flow habitats because they're not very hydrodynamic, and this drag's going to be pulling on them a lot. Whereas if you have a fusiform, narrow, hydrodynamic fish, they won't be expending nearly as much energy to occupy these high-flow areas. And so these deep-bodied fish are restricted from these high-flow areas. And I saw this in a few other traits, such as anal fin length, body depth, dorsal fin length, head length, pectoral fin length, and pelvic fin length. And they all showed this decrease in variation as you increase water velocity. I also saw a similar trend when you look at substrate. So this is body depth and proportion of individuals that occupy a structurally complex habitat. So 1 being that they're always found in structurally complex habitats, and 0 being you never are found in structurally complex habitats. And you tend to see this reduction of body depth as you go towards less structured habitats. And so I saw this relationships for a few traits, but not that many. And so why so few? And some of my ideas are that there could be these idiosyncratic selective pressures. So each region has this unique filter that creates a unique relationship between these traits and the environment. However, I tried to avoid that by selecting streams that were really, really similar. But there may be some regional other filter that I did not account for. I just don't know what it is. And Ernst et al. In 2002 saw something very similar with frogs. He saw no consistent relationships in different regions when he tried to do this analysis, looking at the similarity in trait environmental relationships. Another possibility is behavior. If you have a very fusiform hydrodynamic fish, which is optimized for high flow, sometimes they'll behaviorally choose to occupy slow flowing pools, and that might mess up the relationship between traits and environment. I also had some sampling and fieldwork problems, as there always is. So in Benin and Belize, I was missing one ecomorphological group. I didn't catch that many darter-like fish, these fish that are benthic and live in very high flows. I caught only a few individuals in one site in each of these regions. So I probably reduced my statistical power to find this trend. I was also, these two countries had the fewest species in the beginning. They had roughly between 21 and 25 species, but the other regions had greater than 50. And that probably also has a reduction in this statistical power. And then for Cambodia, these streams, I tried to get pristine streams, but they have really overfished their streams. I didn't catch any large-bodied fish. Only, I think the biggest fish I caught was probably this big. And so that may have seriously influenced this environmental trait relationship. And so I wanted to see if there's this common ancestry or maybe convergence across these environment, or a response to common selective pressure in these different fish assemblages. And I used phylogenetic signal to see if this was a common ancestry. So I did see strong phylogenetic signal in both the niche position, their habitat preference, and the traits. And this phylogenetic signal just means that closely related species have more similar traits than expected by chance. And this really is a signal for this phylogenetic niche conservatism, where these lineages tend to maintain their niches over time. And I did see a lot of convergence, you'll see in my next chapter as well. So really, you see a signal of both in these assemblages. It's not just one or the other, but both. So just going over my questions again, I did see that a relationship between functional traits and environmental variables. I did see some traits being consistent, but not always. And then really I see a signal of both a convergence and this phylogenetic niche conservatism. And so our findings really support this idea that the environmental conditions of local habitats influence trait patterns of fishes in a pretty consistent manner. And this water velocity and substrate seem to be this strong environmental filter for these fish assemblages. And they produce these similar trait distributions across all these different regions. And so now I'm going to talk about my second chapter, and this really just focuses purely on convergent evolution. And so that can be defined as this phenotypic similarity between two or more phylogenetically divergent taxa. And many people will take this as evidence for adaptation to selective pressures, especially if they see this to be extremely prevalent. For example, the cichlids and the riflets. And so this begs the question that if you have habitats with stronger selective pressure, would you have stronger degrees of convergence? And so as I showed in the last chapter, we saw that water velocity and substrate structure tends to have a strong filter on these fish assemblages. And so I wanted to look at this. So my hypothesis for these studies is that fish occupying similar habitats will be convergent across all these geographic regions. And that species species groupings from habitats with high water velocity and non-structured substrates will have a much stronger degree of convergence and a reduced functional richness. And so to do this, I had to create these habitat groupings, and I did this by taking the RQL analysis and then clustering based on the environmental scores. And I came up with 7 different groups. And an example of that group is a mid-water species that lives in low flow and high in-stream structure. And so I had 7 different groupings of this. I also arbitrarily grouped species based on their flow preference, and also their substrate preference. And to determine if there's convergence, I used tanglegrams. And so this is two branching diagrams, one being a phylogeny here on the left, and one being a clustering of traits on the right. And so convergence is shown if you are phylogenetically divergent, but morphologically similar, like these dark pink lines. And also to test for the degree of convergence, I used Staten C1. And so this is just the minimum distance between the extent the taxa divided by the maximum distance of any of their ancestors. And this ranges from -1 to 1, and -1 being the most divergent they could be, and 1 being the most convergent they can be. And so looking at my first hypothesis, I did see a ton of convergence. 70 out of the 197 species were convergent with at least one other species, and that's almost 36 of the species that I analyzed. And here's my tangogram. So the bottom is the phylogeny, the top is the traits. I'm just showing here an example of how I found it, how many species were convergent. And so some examples of this. These are species that tend to live in benthic habitats that are in high flow. And you can see they all look very similar, and these are all from different orders. Yet they look remarkably similar. They all have this fusiform compressed body shape. They all tend to have these larger pectoral fins that they can use to create negative lift, so they won't slide down from the substrate. And one of my favorite examples was this between a hatchetfish and a leaping bard, and they also are in two different orders. And they tend to have these large pectoral fins that they can use to launch themselves out of the water away from predators. And they're also surface feeding, so have these superior mouths to feed at the surface. And so looking at my second hypothesis, I wanted to see if you have higher selective pressure, so higher water velocity, would that have higher convergence strength and lower functional richness. And so I see this, so these are plots for C1 for water velocity and functional richness for water velocity. So in low water velocity, we tend to have low degrees of convergence and higher degrees of convergence in high water velocity. And we tend to have much lower functional richness in these high water velocity habitats. And I also expected to see something similar with non-structured substrates. So I expected to see higher convergence in non-structured substrates and lower functional richness. And that's what I saw. We tend to have higher functional or C1 values in the medium and low versus the high, and much lower functional richness in high structural complexity habitats. And so this shows that the water velocity and substrate complexity are these strong filters, and they tend to restrict the traits that can occupy these certain habitats. And so these high selective pressures tended to reduce these functional richness. And I like to think of this as in the adaptive landscape. You have strong selective pressure, you should have these tall, narrow peaks. So you have this really narrow range of morphologies that can persist in these particular areas. And if you have a low selective pressure, you have this really broad peak. So there's you can have much more morphological variation in these low selective pressure habitats. And we saw that widespread convergence in these microhabitats. And although we did also see that there's strong phylogenetic signal, and just like the last chapter, it suggests that there's phylogenetic niche conservatism and convergence signaled in both of these assemblages. And water velocity and structural complexity reduce and increase these degrees of convergence. And so convergence niches seem to be fairly predictable among streamfish, with species from widely divergent lineages having evolved in response to similar adaptive peaks defined by these similar selective pressures. And so this suggests that evolution, this structuring of these assemblages, is deterministic rather than just purely random. So now we're going to move on to my third chapter, and this really is looking at the functional and phylogenetic structure of these assemblages, and I'm trying to see if the structure is similar across all these different regions. And I'm really only focusing on two community assembly processes when I'm looking at this. Environmental filtering, which is that idea that the traits are restricted and certain environments restrict the traits of species so they cannot persist in a given environment. And limiting similarity is this idea that species will avoid competition within an environment by segregating their niche. And in this case, I'm looking at niche segregation via morphology, so morphological differences. And no real general rule or pattern has been proposed for this. And this is probably because a lot of work has been done on plants and microbes and not that much done on mobile organisms and vertebrates. And so the research is really biased in one direction. And probably each taxon tends to have these assemblage processes affecting them in different ways. And there's not that many large studies that compare these processes across different evolutionary lineages and zoogeographic regions. And it's hard to do compare across studies because everyone uses different methods, different scales, and different ways of measuring traits and different null models and all kinds of different methods. So it's hard to compare and get these general rules. So I'm going to be using these phylogenetic and functional dispersion methods to try to get at the relative contribution of limiting similarity to environmental filtering. And so just to give an example of how this is done, I've created a theoretical fish assemblage here. And so this is just the morphospace of a fish assemblage. So if environmental filtering is acting on these species, it should restrict the traits that within that habitat. So we have habitat A here, and environmental filtering is acting on that. You should have morphologically similar species compared to the rest of the species there. And so it restricts the traits that can occupy habitat A. Whereas if you have limiting similarity and they're trying to segregate in their niche, they should be morphologically different. And so the morphology would be over-dispersed compared to the random. So here you can see that you have now morphologically different species and they're segregating their niches in that way. And these assemblage processes are really dependent on scale. So at the regional scale, you expect to see things like speciation, extinction, and dispersal, and then regional environmental filters acting on these regional species pool. But at the local scale, you have more species interactions and local environmental filtering, and local environmental filters influencing this local species pool. I'm also looking at this stress dominance hypothesis in this chapter, and that's just the idea that functional diversity should be reduced as environmental stresses increase, and this is probably due to stabilizing selection should also increase with this environmental stress. Although this idea has had pretty inconsistent results, and I think fish are going to be a great model to do this. And as we saw before, water velocity and substrate structure tend to have this probably has a strong influence on the stress on these fish assemblages. So my first objective of the study is to evaluate these phylogenetic and functional dispersion patterns across all these zoogeographic regions at the microhabitat and stream reach scale. And my idea is that environmental filtering should be the dominant process at both scales, and then overdispersion and limiting similarity will probably be more important for tropical regions. And I also wanted to test this relationship between the environmental variables and the functional trait metrics and phylogenetic metrics. And I expect that functional diversity would decline as you increase water velocity, and that it would increase with environmental variables that have less stress, like increased water depth and structurally complex habitats. So again, I'm looking at these all these places, and now I'm working at two different scales, both the microhabitat and the stream reach scale, which is just this like 200 to 500 meter section of the river that I sampled. And so again, I structured grouped my traits into three different groups. So traits associated with habitat use, traits associated with diet, and then all the traits combined. And I expect that environmental filtering would influence habitat-associated traits much more than diet traits, and that limiting similarity should influence diet traits as a resource acquisition more than habitat traits. And so I'm hoping to see different trends between these trait groupings. And I'm calculating functional metrics using three metrics. Raoult's quadratic entropy, which is just the sum of the pairwise distance weighted by their relative abundance. Functional richness, which is the convex hull. Think of it as the area of morphospace they occupy. And mean nearest neighbor distance, and that I'm doing using both Euclidean and Gowers distance, depending on the data set. And for phylogenetic dispersion, or the phylogenetic metrics, I'm using mean pairwise distance, which is just the phylogenetic distance between all the species, and that's the average of that. And then the mean nearest neighbor distance, which is the distance between a species and its nearest neighbor, and the average of that of all species. And to see if these metrics are overdispersed and underdispersed, I have to calculate this standard effect size. And so this is just what you observe in your community compared to a random model. And so if your your observed is less than the random, you should find these negative values where you find underdispersion. And so that would be like community A, where you have environmental filtering. So it should indicate environmental filtering. Where positive values indicate overdispersion, which would indicate limiting similarity. And so to get these simulated communities, I created null models. So if we go back to that theoretical assemblage, here's the observed community. And what you can do is randomly select species from this and create random communities. And then you can compare the functional metric values from your observed to your random to see if they're over or under dispersed. So I'm using two models, independent swap, which randomizes the species abundances, and taxon label, which randomizes the species traits. And so looking at my first objective, I wanted to see if there's consistent patterns of all these. And this is a kind of an overwhelming plot. So just to orient you, all the three metrics are on the rows. So this is functional richness, mean nearest neighbor, and Raoult's quadratic entropy. And then the columns are the different countries. And then each of the bar plots are the different traits. So you have habitat, trophic, and all traits. And so the dark black, or the black is significant underdispersion. And the light gray is significant overdispersion. Gray is no different from random. And so one of the general pattern you see here is that underdispersion is much more prevalent or dominant than overdispersion in these communities, no matter which region you look at. And I only really saw some overdispersion in Belize and Cambodia. In Belize, we saw this in the trophic traits, which suggests that they're separating on this trophic axis, so they're separating resource acquisition. In Cambodia, we saw it for all trait methods. And I also looked at the stream reach scale, and most of this showed underdispersion at the stream reach scale. And this is not from that; it's the same plot as before. And so I only saw overdispersion in Cambodia and Belize, and I was really expecting to see this more, in these other tropical regions like Brazil. And some of the reasons why maybe there's not actually any limiting resources there. And so you need limiting resources for competitive exclusion to occur. And this may be because these are pretty small streams. Every time it rains, they have this huge hydrological disturbance. And so this may be keeping the population below carrying capacity. So they're not ever reaching this point of competitive exclusion. Another reason may be just the size of the regional pool. And I think the size of this regional species pool may affect the likelihood of you detecting overdispersion. And what you notice is there was a lot of dark gray. There's a lot of no difference from random. And so this suggests that there's stochastic factors happening or influencing these assemblages. Or it could be that there's contrasting patterns of traits. So if you have one trait showing overdispersion and one trait showing underdispersion, and you combine them, you'll have an overall no dispersion pattern at all. And I try to control for this by using these different trait groupings. So looking at diet traits versus habitat traits. But I still see a lot of just random, no difference from random. And another one of these reasons may be that there's this idea of many to one between function and trait. So an example of this is that this suckermouth catfish, this lorikeet, its mouth, they use it for grazing algae and detritus off of rocks, but they can also use it as a suction cup to stick to a rock so in high flow areas so they can maintain a position. So in this case, this trait could be considered both trophic and a habitat trait. And so it's hard to distinguish between the both and what dispersion pattern may be influencing that trait. And other past studies have shown that stochastic processes tend to increase with disturbance. And so if there's frequent hydrological disturbances in these streams, you may just be seeing these stochastic or no difference from random. And here is my phylogenetic dispersion. So again, the black is the significant underdispersion, gray is significant overdispersion, and dark or light gray is overdispersion, and dark gray is no different from random. And so I see that Benin, Brazil, and Cambodia tend to have lots of underdispersion, phylogenetic underdispersion. That just means that species, these microhabitats tend to have species that are more related than expected by chance, whereas Belize and the United States tend to have the same amount of overdispersion as underdispersion. And overdispersion is just that these species tend to be more unrelated than expected by chance. And some people take this as community assembly processes, so overdispersion would be linked to similarity. It's not good to interpret it this way, and this metric this way is not really suitable for detecting assembly processes. But it can give you some insight to the evolutionary processes. So I created this plot here where you have phylogenetic dispersion on the x-axis, and then functional dispersion on the y-axis. In this case, I showed Rao's quadratic entropy. And I divided this into quadrats, so by 0, so you can have negative and positive values. And so, quadrat A would be overdispersed functionally, but underdispersed. Don't touch the screen. Overdispersed functionally, but underdispersed phylogenetically. So you tend to have this niche segregation between related species, and you have this divergence in these morphological divergence in these microhabitats. Whereas B, you have overdispersion in functionally and phylogenetically, so they're more unrelated and they're functionally unrelated. And so you would have this niche segregation among unrelated species. And then you can have section C, where you're both phylogenetically and functionally underdispersed. So this would indicate some kind of stabilizing selection and have a signal of phylogenetic niche conservatism. And finally, D, you would be phylogenetically unrelated but morphologically similar. And this would indicate convergence within these microhabitats. And one thing to just remember, these are microhabitats within a stream reach. And so you can see that there's a lot more of this C, this signals of niche conservatism when there is a convergence within a stream reach. So when I was looking at this is my second hypothesis for the study. So I expect that functional diversity would decline with increased water velocity and deep increase with water depth and structural complexity. So I found that functional richness was reduced by water velocity and increased by depth, and also saw that Rao's quadratic entropy and nearest neighbor distance increased when you had structurally complex habitats. And so that supported the hypothesis that I was looking at. So my conclusion of this chapter are that environmental filtering was relatively more important than species interaction, and it seems to be this universally important assemblage process across all these regions. And I found some support for the stress dominant hypothesis, where environments with higher stress tended to reduce the functional diversity. And I found higher instances of functional trait underdispersion coupled with functional or phylogenetic underdispersion. So signals of niche conservatism, and probably through stabilizing selection. So just an overall summary of my dissertation. I looked at these 5 zoogeographic regions to look for, using these consistent methods to look at convergent evolution, similar patterns of convergent evolution, and similar patterns of functional trait and phylogenetic structure. And I found that these environmental filters seem to be pretty predictable. Fish assemblages tend to have a predictable response to these environmental filters. And then we found that convergent evolution was very prevalent, suggesting that these key repeated mechanisms influencing the trait evolution and the assemblage structure of these fish assemblages. And that water velocity and substrate structure strongly influence the stream fish assemblages, and in a predictable way. And if you are able to predict, use this to predict how they influence fish assemblages and the evolution of fish, this can greatly improve some biological fields, such as invasion and restoration ecology. If you're able to predict if a fish is able to persist in a given microhabitat or not, you're able to predict if you put the fish there, if they'll persist there, or if they'll go and invade a given habitat. And I think my study really helps with predicting what would happen if you have habitat alterations. For example, if you remove all the in-stream structure by removing the riparian zone, removing the wood out of the stream, or altering the flow regime, it would have huge detrimental effects on the functional diversity of these stream fish. And finally, if I can get on a soapbox, it was kind of annoying in the study is the lack of natural history information. I really wanted to add other niche dimensions to the study, such as natural history, life history and defense and diet, but there wasn't that much information for many of these species. I think that's one thing that we need to do more studies of, is just this basic natural history. And that'll give us a better idea of what's going on in a lot of these assemblages. And also, a lot of studies just know the correlation between environment and traits, but we don't actually know how these traits perform along these environmental gradients. And I think needs to be a lot more studies on these trait performance. But this is a daunting task because there's so many environmental factors that can influence these relationships. And with that, I would like to acknowledge a bunch of people. This is a lot of work to do. My advisor, Dr. Weinmiller, and my committee members, Dr. Conway, Dr. Blackman, and Dr. Fujiwara. And I do a lot of field work for this study. And David Symes helped me out a lot with many of these places. Also, Chad Bays, he spent a lot of time with me, which is surprising, in New Jersey, which was one of the worst places I did field work. And it was the most painful. And then I had so much field work help from many, many people. And I also would like to thank my lab members, especially Eduardo and Fred, that helped me a lot with R and statistics and just talking through some of these statistics. And then Dr. Alphonse and Dr. Jansen, they really went out of their way to help me when I went to Benin and Brazil. And especially Dr. Jansen, he really, really helped me out many times. I went out of his way to help me out. And to my funding sources and to my family for supporting me throughout this whole time for many, many years, and to my friends for the fun times here. And a lot of people have a slide of pictures for their friends, but I probably shouldn't do that. They're not so great. So I'll leave it with this. Thank you.
Well, we can take any questions during this public portion of the event, if you have any.
Yeah.
So did you try to standardize those water velocity measurements across the geographic regions? Because, right, so for example, if you're sampling water velocity at like the temperature that pile flow versus 50%, you're going to come to different answers from fish or velocity.
No, so these are all microhabitat, not like discharge. It's just meters per second. But no, I didn't standardize it at all. But a fast flowing is a high water velocity here is the same as very high. I don't think the number changes necessarily. I did have some streams that were slightly higher flow in general compared to others, but I didn't standardize it. No.
So, congratulations on your great presentation and doing all this work. You started by talking about MacArthur's ideas that these classical patterns will lead to these rules that let us know how ecology works. And you also mentioned Lawton, you know, in his paper, where he says the quality is a mess. So after you've done all this,
Which way would I lean?
What are your thoughts about cleaning up this mess?
I think it's still, I did not fix any of the mess. I don't think. I did see some patterns, but I think it is, I'm not sure how you can get these very predictable patterns. There is just so much variation. But yeah, I'm kind of in between the both. I can't go fully towards MacArthur's that we're going to find these perfect rules, but at least we will find these general patterns of, you know, in high flow, I know which species will probably be there versus low flow. There are some predictability to this, if that answers your question.
Yeah, it does. I had another one go with quick, if you don't mind.
Sure.
You know, you talked about convergence, you have that ABCD plot, but I didn't remember what.
Yeah.
And, you know, that's an example, was I'm wrong, of, you know, a pattern and then making a, sort of post hoc hypotheses about the pattern, like the what gave us convergence.
Yeah.
So why not just, you know, actually look at convergence, which was the different evolution of similar traits or similar functions, and you know, map those traits on the biological.
So I did when I looked for convergence in these microhabitats. That one chapter is where I used the tangogram to show, you know, phylogenetically divergent having similar traits across all these microhabitats. But that plot was both microhabitats, and so you're looking at within a region, so not across regions. And what I didn't mention in that chapter is that within a region, you don't see much convergence, maybe one or two instances of convergence within a region, well, at least my small streams. But across regions, you see lots of convergence. And so there, I didn't expect to see much convergence anyway, because it's within this microhabitat within a region, like within like a 200-meter stretch of the stream. Yeah.
That's good stuff. I'm curious how, so it sounds like the regional species pool kind of important in terms of so I'm interested in what you think about how applicable this would be if we were to look at sort of younger biogeographic regions like Canadian Arctic or something. Would you expect to see some of the same filters emerging in terms of what's predictable?
Yeah, I would say the same filters, but as far as the regional species pool, I think it was that you should find more underdispersion in these younger pools, or at least some people hypothesize that because, or you have this rapid radiations and like more recent rapid radiations in these these newer regions, I guess. And so you'd have more similar species, so you'd have a lot more underdispersion than older regions. And I didn't really see that. I kind of saw the opposite of that. And so I expected to see more newer regions having more dispersion. But overall, so I'm focusing really on these microhabitats and small scale. Like when I say a region, it's still 500 meters of stream. It's not a big, a regional pool. And so I would expect these processes to be similar across these different regions. And the regional pool is really important. I'm thinking why I saw this lots more underdispersion in these older pools like Brazil is because I have this huge regional, functional diversity. It's massive. If you think of this space, it's huge space. And then I find this little bit of that space, like when there's filtering. And if I had a smaller pool and they had the same amount of filtering, the significance would be much greater for that huge than it would be for that huge species regional pool than it would be for that smaller regional pool. So I think there's I haven't seen anyone do any methods or way of standardizing the size of the regional pool to account for this really.
No other questions? We'll conclude there. Thanks. Thank you.
I think cut out.
Really,
Just let me know when drinks are happening.
Yeah.